Hey, bio folks, hopefully you made it through day one and all the Schoology goofy issues that were going on. Anyhow, we did viruses part one. Today we're going to do a quick, short look at viruses part two. And again, some of this is setting us up for biotechnology. And we'll discuss that as we get going. Anyhow, again, after you're done here, there'll be a quick little homework quiz, whatever you want to call it, to do. And hopefully we have no issues. Hopefully this video goes as viral as yesterday's. Anyhow, yesterday was really just looking at bacteriophage and not so much animals, although the similarities can be drawn there, certainly to some extent. But it was really just getting the basics of what is a virus, just genetic material wrapped in a protein coat, and the two stages of lytic and lysogenic. And we talked about prophages and stuff. And like I said, again, a lot of it wrapped around bacteria. Animals are different because, as we ended with yesterday, you're looking at two kind of key variables about infection with animals and viruses. And one, of course, is a DNA or RNA. Second is a single-stranded or double-stranded. So a couple of charts that I have here from your book, and you can look at them also, and you can see here that you have some double-stranded DNA, some single-stranded DNA, some double-stranded RNA, some single-stranded RNA, etc. And then the retroviruses at the bottom. A couple things, what diseases does it cause? That certainly is interesting. But part of what I'm looking at here is, does it have that envelope that we talked about? That envelope that is at least part from the host, which then does a few things. There's a protective measure, there's an immunity measure, there's a continuing to propagate the virus without lysing the host like you would in a lytic cycle and stuff along that line. So that will be part of the discussion of then how these viruses can continue to infect or have that time. Probably a time to remind you that antibiotics have nothing to do with viruses, because as we kind of had to look at yesterday, viruses are not necessarily considered living. Antibiotics are for bacteria and other pathogens and living pathogens. And that's really what happens with viruses then, is that it comes down to your immune system responding to recognizing that virus as not being a part of you. And the problem with that, and this is where some of these things get a little dicey or nasty, is that first infection that you ever get, and this is why you take vaccines. Vaccines are to trick you and trick your immune system into creating the antibodies, because the first response can go anywhere up to 2 weeks. You're looking at maybe a 10-day, 2-week response before your body has had the chance to build an antibody that can attack that viral infection and destroy it. And hopefully, depending on the virus, in that 2 weeks, you're not to that point where you have already succumbed, or you're past the point of no return as far as the infection in your body. The second time, then, anywhere 1 to 3 days, you can have those antibodies going and knocking things down. And that's part of the key to it also. Anyhow, so one of the things I wanted to point out here, besides looking at some of the viruses, like the adenovirus, herpes virus, the gift that keeps on giving, and stuff like that, is if you look, the majority of RNA-based viruses also have an envelope. Not all, but the majority. Where a lot of DNA ones don't have the envelope, and there's some differences too. For example, the herpes virus does have the envelope, but it doesn't last. It gets out in the cytoplasm and doesn't last and stuff. So looking at that single-stranded or double-stranded, you see a lot of envelope surrounding the RNA stuff, including the retroviruses. Okay, so let's talk about those envelopes a little bit. Many of the viruses, as we saw, that infect animals have a membranous envelope. the viral glycoproteins on the envelope bind to specific receptor molecules. Really, you have two ways in. They're either going to bind like receptors, as we've seen before, or they can be engulfed by endocytosis and brought in. Okay. Anyhow, some of the viral envelopes are formed from the host cell's plasma membrane as the viral capsids exit. Others form as the host nuclear envelope are replaced by an envelope made by the Golgi and stuff. So it can be in that endoplasmic Golgi apparatus extravaganza, either or. What you see here, and I got this picture from your book, and they're really not showing here the binding entry, but it enters into the host cell. And then what you see here is, of course, the capsid is broken down by enzymes, and here's our viral genome. And using the cell's machinery then makes a template of its stuff. And what you see here is both things are copied. Not only do you continue to copy the original genome so you can insert that in, but also your complementary strand is also copied. And you can see in this case here that a lot of those glycoproteins, the capsids are made, and a lot of the glycoproteins are made in the endoplasmic reticulum and transported. Now notice that they are attaching to the outside of the plasma membrane, so that as the capsids assemble and take the genome, that the new virus is then budded out and forms. And of course, part of that is to defeat the purpose of having that lytic cycle where they would break down the cell itself. Not that that doesn't happen in animal cells, but this is in discussion of how the envelopes work. Okay, now let's talk about retroviruses. Retro means backwards. And so sometimes, how do I want to say this? Sometimes as you look at stuff with these, as I go back to this envelope, sometimes you'll have viruses bring in some of their own stuff to help in the host cell. You know, it's not that the host cell may always have all the enzymes and everything that is brought in. And that's one of the things that happens here with retroviruses, is there's an enzyme reverse transcriptase. And that is not something that your body has. So that is something, as we're going to look at a picture in a minute, that you're going to see helps catalyze the synthesis of the RNA, but is not something necessarily is from the host, is what I want to say. It essentially has to be brought in by itself. Anyhow, the DNA that acts as a provirus, which is like a prophase, but in the animal cell, becoming part of the host, and much more dangerous. And this is what I wanted to allude to earlier when we were talking about prophage versus reverse transcriptase retroviruses and a provirus. The provirus is then with you forever. In bacteriophage, the prophage, as soon as the host bursts, then it's done. But you never lose the DNA in the retrovirus. Okay. So broadest variety of RNA genomes is found in viruses that infect animals. Retroviruses use transcriptase reverse to copy their RNA into DNA. Okay, and we mentioned that HIV is a very dangerous retrovirus. And here's a picture that I got from your book, and you can see that it's being brought in endocytosis or binding to certain sites. And the capsid breaks down, and there's the host cell. And then you can see that the viral RNA, with its reverse transcriptase that it brought in, then makes an RNA DNA hybrid, which then makes the DNA double-stranded nucleus, which then gets inserted. And then is always there to reproduce and send off the new virus. Same thing, copying making messenger RNA to make both the glycoproteins for the outside of the plasma membrane and the reverse transcriptase and the viral genome stuff, and then you can butt off. But this is the key: you never lose that provirus. Okay, that's just there. Okay, so and here you get to see that budding on a micrograph. Okay, unlike the prophage, the provirus remains a permanent resident of the host cell. The host RNA polymerase transcribes the proviral DNA into RNA molecules, and then the RNA molecules function both as messenger RNA of the viral proteins and as genomes for the new virus particles that are going to be released from the cell. Okay, so where did viruses develop? It's a good question. There's some discussion. They don't fit our definition of living organisms as you looked at. Viruses replicate only within the cells, so the thought is they developed as bits of cellular nucleic acid, and how they look at that is you look at plasmids and plant and bacteria and stuff that are their own circular DNA stuff. Transposons, those movable parts of the genetic complement that are small and mobile. That's probably the development, as you had these essentially free genetic material. They evolved into a way to have a protein coat so that they could remain viable and not get chewed up by enzymes and stuff in the environment until they got passed on to the next host cell. Okay. Wrap it up. Is there other infectious agents other than a virus? Certainly are. There are fungi. I'm a fungi. There are viroids, which are small circular naked RNA that infect plants, causing abnormal development and stunted growth. Few hundred nucleotides long, replicate using the plant enzymes and cause errors in their regulatory systems. Any single molecule can be an infectious agent. Anything can be. Okay, whether it's a protist or a bacteria or a fungi, I'm a fungi, a virus. You can also have prions, which are infectious proteins. And you may not remember, but for a while there was a problem with beef in the European continent because of mad cow disease. And that turns out to be an infectious protein. They're slow acting, many years to incubate. So by the time they start showing what they are doing, many people are infected. And they convert normal proteins themselves. I'll go back to the link in a minute, but I stole this picture from your book. So you can see one of those prions comes in, and really they start infecting the other normal proteins and it become a dangerous infectious agent. What I have here for you, if you click on that link, is a link to the CDC where you can read about different prion diseases and what they do. Okay. Anyhow, that is the quick and informative, I hope to some extent, virus video, second part of the virus video, just kind of talking about animals and both single and double-stranded DNA and the envelopes and how they work. There will be a little bit of a homework quiz of sorts, so please give that a shot. We'll get more and more into this virus stuff as we go and talk about biotechnology, because again, a lot of those enzymes that we talked about with bacteria fighting viruses and stuff are the things that are used to do more biotechnology. Anyhow, stay safe, help this go viral, and we'll talk to you soon.